In this video, I will attempt to review fatty acid metabolism and also focus on some of the very high yield aspects of fatty acid metabolism, and then we'll also take a look at some of the disorders associated with fatty acid metabolism. Now, fatty acids need to get into the mitochondrial matrix in order to undergo beta oxidation. Now, the long-chain fatty acids require the carnitine shuttle to move the fatty acid into the mitochondrial matrix. But fatty acids cannot cross the mitochondrial membrane, especially the inner mitochondrial membrane, and that is why we need this carnitine shuttle. Now, the carnitine shuttle begins with the fatty acid reacting with ATP, and the enzyme fatty acyl-CoA synthetase in the outer mitochondrial membrane. And this enzyme, fatty acyl-CoA synthetase, converts the fatty acid to fatty acyl coenzyme A. The next enzyme is carnitine palmitoyl transferase 1, or CPT1. And note that there's also a carnitine palmitoyl transferase 2 in the inner mitochondrial membrane. And then we have another enzyme called carnitine acylcarnitine translocase on the inner mitochondrial membrane. So once the fatty acyl-CoA is formed, it is then passed on to carnitine palmitoyl transferase 1, CPT1. And what this enzyme does is that it replaces the coenzyme A on fatty acyl coenzyme A with carnitine to form fatty acyl carnitine. Okay. Now, this fatty acyl carnitine then crosses the inner mitochondrial membrane with the help of carnitine acyl carnitine translocase, as you'll see here. And the fatty acyl carnitine then finds itself inside the mitochondrial matrix. This fatty acyl carnitine needs to be converted back to fatty acyl coenzyme A. And so the fatty acyl carnitine goes to carnitine palmitoyl transferase 2, or CPT2. And here the carnitine is removed and the coenzyme A is added to form fatty acyl coenzyme A. It is this fatty acyl coenzyme A that will undergo beta oxidation. Now, beta oxidation consists of a number of steps. It begins with fatty acyl coenzyme A, and the fatty acid is converted to these various intermediates, ends up as beta ketoacyl coenzyme A, which is then converted to fatty acyl-CoA, and this fatty acyl-CoA is, of course, 2 carbons shorter than the original fatty acyl-CoA that we started off with. In addition to this, you also get the release of acetyl coenzyme A, which then goes off in the TCA cycle to generate ATP. Now, the very first enzyme in beta oxidation you'll see here is the enzyme acyl-CoA dehydrogenase, which I will talk about in the next slide. So the fatty acyl-CoA that remains goes back to the beginning of beta oxidation and goes through yet another cycle. So that very first enzyme I mentioned earlier, medium chain acyl-CoA dehydrogenase, can present as a deficiency in some infants, and they do not have the enzyme, which means they cannot carry out beta oxidation. And you remember that each fatty acid has a methyl group at the one end, a carboxylic group on the other end. So because in the absence of this enzyme, the fatty acid cannot undergo beta oxidation, and instead it undergoes omega oxidation, to form dicarboxylic acids, and these dicarboxylic acids are about 8 to 10 carbons long, and these can be detected in the urine of these infants, and this is a diagnostic feature of this deficiency. And so in those patients, you have to decrease fat intake and increase carbohydrate ingestion. The next question that arises is what happens to these to very long chain fatty acids? They cannot go to the mitochondrial matrix for beta oxidation, so instead they convert it to octanol-CoA in the peroxisomes. As the name implies, these are 8-carbon fatty acids. And once these are broken down to the octanol-CoAs, it goes back to the mitochondria for beta oxidation, and I must point out that going through this method, the very long chain fatty acids, they do not produce as much energy as the medium chain fatty acids. So now let's take a brief look at some of the disorders of peroxisomes. Okay, and here we have a very important disease, high yield Zellweger's syndrome, and in this particular syndrome, there is a defect in the peroxisome biogenesis, in other words, synthesis of peroxisomes. So they cannot metabolize these very long chain fatty acids to the octanoyl CoAs that I showed you earlier. And so they accumulate, and this affects all of the tissues. Another one is the X-linked adrenoleukodystrophy. And in this particular disorder, again, the very long chain fatty acids will accumulate. They accumulate in the blood and in the various tissues. And this disorder is associated with small adrenal glands. And in addition to that, there is also a decreased myelination of the neurons in the brain and the spinal cord, leading to a neurological disorder. Another very important consideration is the metabolism of phytanic acid. This is derived from chlorophyll, and its concentrations are very high in the meat and milk of ruminant animals. So phytanic acid has an even number of carbons, but it has to undergo alpha oxidation to form a 17-carbon pristanic acid, which is then converted to propionyl coenzyme A and then to succinyl coenzyme A, as you might have seen in other situations. So this alpha oxidation converts phytanic acid to pristanic acid. And some individuals are born with a deficiency in the ability to carry out alpha oxidation, and this results in an accumulation of phytanic acid, which can lead to a disease which we call Refsum disease. So remember, I said Refsum's disease results from the absence of alpha oxidation of phytanic acid, and therefore phytanic acid will accumulate in these patients. And this can lead to a neurological disorder, which we describe as Refsum's disease. And so in these patients, we need to restrict the dietary ingestion of green vegetables, as well as ruminant milk and meat, in order to decrease phytanic acid levels in these individuals. Now let's take a look at the oxidation of odd-chain fatty acids. Remember, as I showed you in the previous case, if you have a fatty acid with an odd number of carbons, it leads to propionyl coenzyme A, which is converted to methylmalonyl CoA, which in the presence of vitamin B12 or the enzyme methylmalonyl CoA mutase is converted to succinyl CoA, and which then enters the TCA cycle. Now, with regard to fatty acid synthesis, this is carried out by an enzyme complex. It's a complex of 7 enzymes called fatty acid synthase. And these enzymes surround a protein called ACP, or the acyl carrier protein. And this complex has 2 arms, what we call the SH arm, which is built up from the vitamin phosphopantothene, which is directly attached to the acyl carrier protein, and it carries the intermediates to the various enzymes. The other arm is known as the KS arm, which also carries some of the intermediates of fatty acid synthesis. Now, what you need to remember is that the first enzyme in fatty acid synthesis is acetyl-CoA carboxylase, which then converts the acetyl-CoA to malonyl coenzyme A on the SH arm. And very important point for you to remember is that malonyl CoA inhibits CPT1 in the carnitine shuttle. And so the this process is followed by a reduction of the fatty acid with NADPH, followed by a dehydration reaction, and then once again another reduction reaction with NADPH, which of course comes from the pentose phosphate pathway. Okay? And eventually you get chain elongation until you get a 16 carbon fatty acid, which is palmitate or